How's it going, guys? Hope you're doing well. Hope you're having had a great weekend. So this week we're going to be talking about Mendel and heredity. And you may have heard of heredity, but I know that some of you are like, "What the heck is Mendel?" Um, well, we're going to talk about that, and in this is all covered in chapter 11. So in case you wanted to look in your book too, if something's really not making sense, even after like our Zoom meeting or any of the videos. So yeah, let's get started. So this is a picture that Mr. Cervantes put in these notes, and so this is Mr. Cervantes obviously on the right, and his wife here on the left. So the questions are for you to make 3 predictions about what you think their kid, or well, we now know that it's going to be a daughter, or is a daughter. What she'll look like. Why did you make these predictions, and what additional information would be helpful in making this prediction? So I'll give you guys a second to do that. You can pause the video and think about that, and then get going when you're ready. So I'll be interested to hear some of your predictions of this at our Zoom meeting. So let's get started. So what is heredity and what is genetics? Heredity and genetics are just the study of how traits are passed from one generation to the other. So some goals today is being able to predict how traits will be passed on from one generation to the next, and you do that using what's called the Punnett square, and we'll talk about that a little later. And be able to explain how traits are being passed on based on our knowledge of genes and chromosomes. So let's imagine a scenario. So there's a genetic disease that could kill you when you get to age 40. You don't know if you have the disease or not. And if you do have the disease and you have children, you could give that disease to your children. Now, there's a couple questions. Would you want to know about your impending death? So if you had this disease, would you want to know that you had it? Or would you let it be a surprise? So that's that age-old question of like, if you knew exactly when or where you would die, would you want to know, or would you rather live your life not knowing that? With this knowledge, so if you did know that you had this disease, would it change your decision about having children? So you know that it's living in your genes, so you could potentially give this disease to your children. Would you have them? Or would you want to know the probability of passing it on? Well, that's cool because in today, currently, we have doctors or genetic counselors that can actually sequence your chromosomes and look at your chromosomes and tell if you have any diseases or potential genetic diseases or not, which is pretty cool. So the disease that we're actually referring to in this example is called Huntington's disease. Some of you might have heard of it before. So here is actually a chromosome. You can see this chromosome arm here, and it's actually your chromosome number 4. And here's a gene. So you have this gene, which is just called a Huntington gene. Here it's labeled HD. And again, a gene is just a section. So this little blue section is what the gene is, this little section of the DNA. Right, and I'm going to eventually transcribe that DNA into RNA. Eventually, that RNA is going to get translated into amino acids. And normally, under normal conditions, I'm going to have about 36 of these amino acids to give me this functioning protein. But I can have a mutation or an abnormal Huntington mutation, and that's going to give me more than 36. And that results in what's called Huntington's disease. Eventually, that would kill you. Huntington's, what I remember about it is that it's similar to Parkinson's in some ways. It affects your brain. Okay, so a gene versus allele. What is a gene? What is an allele? Well, I think to help us do that, we could start off by drawing some chromosomes. So I'm going to do that here. So I have 2. Remember, if I have 2, let's say these are my first chromosome. So these are actually homologous chromosomes. Homologous. These are homologous chromosomes. Why are they homologous? Well, they have the same gene. They have the same gene. But you're like, wait, Mr. Rubio, no, they don't have the same gene. Look, you can see they clearly are showing different colors, so they're not the same gene. Well, that is the difference between a gene and allele is. So a gene is just, I'll write that here. So a gene, all a gene is a strand, or actually we'll say section of DNA that codes for a specific trait. And that specific, an example of that specific trait might be eye color. Versus an allele are different forms of that gene, or of a gene. And so examples of that would be like green versus blue. So here's kind of the difference there, right? So you can see that I have the same gene because, right, it's just a section of my DNA that codes for a specific trait. So both of these genes, I'm going to say code for eye color, so that the same gene, but they're different alleles. Why are they different alleles? Well, because they result in a different form. And that different form is going to result in different trait, which is either green eyes or blue eyes. And why are they homologous? Well, let's do a little reminder of what a homologous chromosome is, right? Well, homologous, because they have the same gene, I don't know why that disappeared. So I get one from my mom, one from my dad, right? So I have two different chromosomes. And they're homologous because they have the same gene, same genes, different alleles though. Okay, hopefully that made it a little clearer. Okay, this is just showing the Huntington's disease, what it actually is. So on chromosome number 4, chromosome number 4 here, you can see this little section, right? A section of DNA is what my gene is. But I can have a mutant gene or a mutant allele, right? Allele, because it's a different form of that gene. And that results, it's going to result basically in a defect in your brain, in the cytoplasms, and it's going to result ultimately in death. Now, the interesting part is that this disease only affects you in like your 40s, so in almost your midlife. So how do you think that, or what are the implications of that? Or in other words, why is that an issue if it doesn't show up until your 40s? Give you guys a second to think about that. So if you took a second to think about that, maybe you paused the video, maybe you didn't, but you might have seen this picture here. Well, the implications are that if you don't know you have a disease until you're in your 40s, you might have children by that time. And if you have children, well, you have a chance that you could pass on that genetic disease to them, right? And so that's one of the big implications. Why, for example, why Huntington's disease or why genetic diseases stick around is because if they don't show up until you've already had kids, well, it's too late. Now I can't stop it from potentially them being carriers or them actually having it. Right, and you can see that here. It's a 50-50 shot if my kid's going to get it or not. But let's take a little step back and let's look at, well, where did this all come from? Where did genetics even come from? And that's from the father of genetics, and his name is Gregor Mendel. Now, Gregor Mendel was just, you might have seen in the Edpuzzle videos, and if there's any point in this lecture or this little short lecture that's confusing to you, I suggest going back over those Edpuzzle videos. They really are good, and I put in some questions, and I even voiced over a couple sections in them to kind of help guide you. But anyway, Gregor Mendel, he was a monk. He loved science, and he actually was really interested in heredity, where in heredity, right, just being how traits are passed on. Back then, people in his day assumed that if you, example, had a black squirrel and a white squirrel, you would get a gray squirrel. Or if you had a black cow and a white cow, it would be a gray cow. Or if my mom was tall and my dad was short, I would be medium height, right? They assumed that it would just be a blending of your traits. And Gregor Mendel actually wanted to explore that a little more. And so before we get into that, let's look at some general notation. So parent generation, or so my parents, are going to be denoted as having a P. The offspring, the first offspring of that, so the first offspring are going to be called F1, and the second generation of offspring are going to be F2. So you could think about that as the parent being like grandparents, the F1 being your parents, and F2 being you. So this goes a little bit into detail of how you actually pollinate pea plants, because plants or pea plants, like a lot of other plants, are actually can self-pollinate. And what does that mean? Well, it just means that they have both female and male parts to them, so they can actually pollinate themselves. So they can have kind of, basically they can asexually reproduce. They can just not have any other plants near them and just keep making more. And this just goes into detail. So kind of the painstaking process you can imagine to create these different breeds or these different forms of pea plants that Gregor Mendel did. So let's look at some of this data. So you have the parental, the pea generation here, and he just had true breeding plants. And by true breeding, all I mean is that anytime I had a plant that had purple flowers, all of its offspring, if I mix them with themselves, so if I put a purple and a purple together, it's always going to give me purple flowers no matter what. And same with these white. If I have these white ones, I'm going to always get that same color. And there's a bunch of other traits, right? I could have seed color, seed shape, and so on and so forth. And so he looked at a bunch of different traits, but we're going to focus here on flower color, and we might do seed color too. So flower color, so let's see. So what he did is he said, well, if I have all these that only make purple and all these that only make white, if I mix them, I should get somewhere in between, right? That's what everyone or most other people think. Well, when he actually crossed them, he got only purple flowers, and he was like, what the heck? This doesn't go along with what anyone's saying. How can I not have something in the middle of them? And then what he did is he's like, well, the only thing I can do is take these purple flowers and see what happens when I cross them with themselves, or what happens when I breed them with themselves. So he took these purple flowers from this F1 generation and he mixed them, and he ended up getting plants that had purple flowers and white flowers. So this was crazy, because all of a sudden the white flowers were back, when in this generation they disappeared completely. Now they're back. And then he did the same thing for all these other traits. And he noticed, or he started to compile a bunch of data. You can see the hundreds, if not thousands of times he did these experiments. He had a lot of time up in that monastery, let me tell you. Anyway, he started to notice trends, and he made these into ratios. And I want you to take a second and see if you can notice any trends in these ratios here. So you might have taken a second to do that. And what you might have noticed is that all of these have about a 3-to-1 ratio. So for example, if I looked at the purple flowers, I had about for every 4 flowers. So for every 3 purple flowers, I had 1 white flower, essentially. Or for every 3 yellow seeds, I had about 1 green seed. And he noticed that trend was continued. So how do we explain this? Well, let's look at, just zoom in on what his crosses were actually doing. So this is the, let me actually write this. This is the F1 generation here. So what he did is he took the P generation, right? He took the P's, the parents, and he crossed them with each other, right? And so they were only, let's say this is seed color. Sorry about that. So let's say this is seed color. So he took parents that were only yellow and parents that were only green, and he crossed them, and he expected that these would all be somewhere in the middle of green and yellow. However, what he actually noticed, right, we saw in the F1 generation, what he actually saw was that all of these ended up being yellow. Why? Why weren't they somewhere in between? Because clearly one of the parents is giving its green allele, right? It's giving its green allele. And you might have noticed this little chart up here, and we'll talk about that in a second. But let's move on. So why aren't they? Let's see. Well, let's look at the F2 generation. So it's my spotlight. Clear. Okay. So let's look at our F2 generation. So now I took those parents that I made in my F1 generation, and these are going to be right. They have a mixture, but these are only, remember, these are yellow. They're yellow, but they still have this green allele in here, right? Just floating around, but somehow they're only expressing yellow. So let's see, I mix these, so these come together, and I get one offspring that's all yellow. I have another offspring that's yellow. I have another offspring that's yellow. But wait, all of a sudden, I have an offspring that's green. So green came back. What do you notice about why this one's green compared to this one? Well, you might notice that there's a yellow in with the green, and you notice that when there's two greens together, it actually makes that color. It makes green. Why is that? Well, this has to do with what we call dominant or recessive, and you might have heard of a trait or something being dominant before, but we're going to explore that a little bit more here in just a second.